Unit 3, Movement into and out of cells. Lesson 2, Osmosis. Objectives. Come back to the objectives by the end of your studying as we're used to. Osmosis. What's osmosis? It is a type of diffusion, but only specific for water. Water also moves from high concentration to low concentration, but in case of water, we call it the high water potential to the low water potential. A solution with high water potential means that this solution is a dilute solution, has less solute in it. Can you see here? The solute molecules are less than the solute molecules on the other side. It has nothing to do with the volume. It's always about the amount of solute inside. And the other side, this is said to be a concentrated solution or a low water potential solution. So water molecules through a semi-permeable membrane, they will move from the high potential to the low potential, which means from the dilute solution to the concentrated solution. Please remember that in diffusion or osmosis, when we reach equilibrium, this does not mean that the particles will stop moving at all. They will just stand still. No, they will still be moving, but the net movement will be equal to zero, because when one particle moves to a side, an equivalent particle will move to the other side. So the net movement will be zero. 1 minus 1 will always be 0. Water is very important. It is the solvent in which we dissolve a lot of solutes. Also, in biology, when we need to excrete urea, the body dissolves, or the kidneys dissolve this urea into water. Any dissolved substances in the blood plasma are dissolved in water, like a lot of nutrients. Also, water diffuses easily through the cell membrane. That's why osmosis is considered a type of diffusion. Okay, osmosis in animal cell. So, animal cells are usually more concentrated, or are considered concentrated, than the pure water outside. And we said that concentrated solutions are low water potential solutions. So this means that they will attract the water to go inside, because the pure water in this case is a high potential water solution. So the water will keep going from outside of the cell to inside of the cell. And because the animal cell does not have any cell wall to protect it, what will happen is that it's going to burst, unfortunately. That's why animal cells are never left in water without protection. When animal cells are placed in a concentrated solution, a solution that has a lot of solute inside, now the water will move from the high potential, which is the dilute solution, which is in this case the solution inside of the cell cytoplasm. The water will move to the outside, because now the concentrated solution outside is a low potential water solution. So the water will move from the inside of the cell to the outside of the cell, which means that the cell is going to shrink because it's going to lose water. Let's take a look on the plant cell. The plant cell follows the same rule. In pure water, it is considered more concentrated, which means that the water will move from the high to the low potential, which is inside the cell. But the plant cell has two privileges. The first privilege is that it has a big vacuole to store water. And the other privilege is the cell wall, which is very strong and prevents the cell from bursting. So the water will start moving inside of the cell by osmosis. What will happen to the vacuole? It's gonna enlarge and enlarge. The cell will swell, but it will not burst. It will become turgid, stiff, as if you inflated a balloon. It's gonna be hard and stiff, which we call turgid. But what will happen when the opposite situation happens, when I put the cell in a concentrated solution. So now the low potential will be outside of the cell, while the high potential will be inside of the cell. And the water will always move from high to low, from dilute to concentrated. So the cell will shrink, but what about the cell wall? The cell wall can't shrink. So what will happen is that there will be a space between the cell membrane and the cell wall. See, this is the cell membrane. See, it's not just sticking to the cell wall as we're used to seeing. Why? Because the water left the cell, and of course the vacuole will shrink, and we will call the cell flaccid. Flaccid means empty and not turgid at all, like an uninflated balloon. The shrinking of the cell membrane away from the cell wall, it's called the plasmolysis. Actually, it's not just the cell membrane. The whole contents of the cell will be moving away from the cell wall. We call this process plasmolysis. See here, now we have a lot of salt ions, so the water will leave, the cell will shrink. The opposite will happen when there are no solute molecules outside, so this means that the high potential became outside, and the water will keep going to the low potential inside the cell until the cell bursts. And you see here, this is a turgid plant cell, while this is a flaccid plant cell. See, this is the cell membrane. It's quite away from the cell wall. Let's answer some questions again. What are features of osmosis? To answer such a question, please look at each one of them and write your own signs before choosing in the bubble sheet. So, he's asking about features of osmosis. Diffusion is involved. That's correct. It is a type of diffusion. Requires cell wall. No, it does not require a cell wall because cell walls are totally permeable. Requires partially permeable membrane, of course. So now it is quite easy to decide. What is the answer, because I have it already. So the answer is gonna be, yes, it's gonna be A. That's correct. The diagram shows apparatus used to investigate osmosis. The volumes of solutions A, B, C, and D were the same at the start of the investigation. After an hour, the solution had moved up the glass tubes. So, the givens are that here inside those tubes that have membranes at their bottom, there are different solutions, and the water was the same amount, the same volume in all of them. Okay? And then after 1 hour, the water moved in all of the tubes. Okay? Which solution was the most concentrated at the start of the investigation? How would I know? I will know because the higher the concentration, this means that it has lower water potential, so the more water it will absorb. So accordingly, the one with the higher concentration will be the one that ended up with the most amount of water, which is gonna be D. Another question. Which diagram shows the appearance of the plant cell after several minutes after it has been placed in concentrated solution of sugar? So if it was placed in concentrated solution of sugar, concentrated solutions are the ones with low potential of water. So the water will move from the high to the low potential. Accordingly, it will be the cell that will turn flaccid. Yes, flaccid cells, they have smaller vacuoles and cell membranes away from their cell walls. Which one does these rules apply on? Yes, A. Why not B? Because B does not have a vacuole at all, and this will never happen. And D has multiple vacuoles, and we learned that in plant cells, we have only one big vacuole in the middle. And this one is a turgid cell, where you can see the cell membrane pushing the cell wall. Okay, so of course it is A. 3 identical potato cylinders, potato that is cut in a cylinder form, were used to investigate water movement in plant cells. Cranberry juice is a red fruit juice that contains natural sugars. The 3 test tubes are shown in the figure are set for 1 hour. And here are the observations of the experiment. At the beginning, the mass of the 3 potato cylinders, and after 1 hour, the mass of the 3 potato cylinders. This is a very important thing to know, that at the header of the table, you have to find the unit. Again, units are very important. Either you are drawing the table or taking any information from the table, you have to mention the unit. Especially in paper 6, while drawing any table, you have to put your unit in the header of the table as shown. So, test tube 1, test tube 2, and test tube 3. All of them started with 25 grams, and these are the differences after 1 hour. Describe the results. Describe, again, is a command word, which means that you only mention what you see. No explanation is required. Test tube 1, what happened? Yes, the potato cylinder increased in mass by 5 grams. As you see here, it was 25 and now it's 30. So this means that it increased by 5 grams. In test tube 2, the mass stayed the same. The next question is asking you to calculate. Calculate means that you're going to do the numerical value and don't forget the unit. Here you go. So, after calculation, the difference will be 6 grams in mass in test tube 3. Explain. Explain questions should be answered in paragraphs. Yes, and you have to mention all the scientific information about the topic given. So explain why the potato cylinder lost mass in test tube 3. We will say that the water moves out of the potato cylinder, and we have to mention the scientific term or the scientific process, which is osmosis. Why? Because more water molecules were inside the potato than outside it, which means that it was moving from the high water potential to the low water potential. So what is this? This is the mark scheme. I put it here so you understand how questions are corrected in Cambridge. Okay, when you see the semicolon, this means that after this is another mark. Okay, and when you see this slash, this means or something else. AW means any wording with the same meaning will be accepted. But this does not apply for the scientific term, of course. Okay, state the name of one mineral ion and give a reason why it is important for plant growth. Magnesium, it's a very important ion. Why? Because we learned before that magnesium is very important in making chlorophyll, and chlorophyll is important for photosynthesis. Thank you, and I hope this video helps you study.